Hello everyone. Uh, we are going to study about the electroencephalography, which we generally know as EEG. So this lecture is for beginners who doesn't have any idea what is EEG, how does it look like. So I just want to give the basic feel of EEG. So we will know what is EEG, how does it get generated, how we acquire EEG data, how does it look like. So for that, we will use two open source like free toolboxes. First, M&E Lab. This is the GUI based M&E Python uh, analysis for the EEG data. We will know how to load EEG data, how to plot raw EEG data, then implement different type of filters, run ICA to remove artifacts, and so many other things. And simultaneously, we will also see how we can do same thing in M&E Python based coding. So actually both are same, but the thing is the first this one is for GUI based, and then this one is the coding based. We will see how does this work and how does this work. And after this lecture, if you have anything to ask, you can ask in the comment box, or you can also follow me on the Blue Sky and ask if you want to know something, if you want to know anything regarding EEG or in general like brain science, or if you are a student wants to know about how to pursue things. Okay, so let's start. So first thing, what is EEG? So EEG, that is electroencephalography. Here, electroencephalography consists of three words. So the uh, term electroencephalography is like we use it. What is this? Like actually, this is if we say what is this? So electroencephalography, three words: electro. Then we have encephalo. Then graphy. What does it mean? Electro means like simple. It represents the electrical activity of neurons, like all those activity originating from the neurons we will call electro. So electrical, these are electrical activity of neurons. What is encephalo? So encephalo mean here head, and what is graphy? Graphy simply means sometime you will see graphy, sometime you will see electroencephalogram. So graphy and gram. So we use this for general things, either to when we want to say something to draw or to write. So what does it mean? Electroencephalography mean we write the electrical activity of our head. Simple. So in basically electroencephalography is the measurement or record of the electrical activity inside our brain. Electroencephalography is the measurement of excitatory and inhibitory postsynaptic potentials. This is important to remember. Later on, we will see in detail what is this excitatory and inhibitory postsynaptic potentials. So for now, you remember EEG is a non-invasive technique. Like we measure electrical activity of our brain from the surface. We don't insert anything inside the brain. Okay, so it is a non-invasive brain imaging technique that provide a continuous recording of the brain's macroscopic electrical activity. Remember that. So it provides the continuous electrical activity of our brain's macroscopic level. So always remember. So it provides the electrical activity of at macroscopic level. Okay. So EEG basically measures the summation of the excitatory and inhibitory postsynaptic potential. Either it can be excitatory or inhibitory postsynaptic. So how does the neurons activate? Like these are dendrites. it receives neurotransmitter and it comes to the body. After summation, there is certain threshold. If this activity cross the threshold, it fires, neuron fires during with the axon, and here it snaps. So after snaps, like we are measuring the electrical activity of the tens of thousands of neurons at the surface level in case of EEG. And mostly these are pyramidal neurons and in cerebral cortex. So the thing is, in EEG, we mostly measure the brain activity of the cerebral cortex, means surface upper layer of the brain, if you can say. It's not possible for EEG to know the electrical activity of the deep brain. Okay. So this is mostly surface level. And these are the surface layer. If you say surface level, this cerebral cortex. So here we measure the electrical activity. Okay. So the electrical activity is typically recorded using the elastic cap embedded with the surface electrode, and we generally use the number of electrode from, like, it can be either 5 or 10, 20, or generally it's people roughly use, like, in case of like 32 or 64, or people also use 124, 124, and it's also possible to 256. So the thing is, these are the common things. You can, like, even there are recordings of channel 1024. So it can be very dense also. So the thing is, commonly we use this much channel. Okay. So what does the how do we measure the electrical activity? So this is our brain. So we put electrode here on the surface of the brain head, and we measure the electrical activity. We use amplifier and we get electrical activity, okay, how it is changing the time. So, and these are, remember, these are in microvolts. It's very tiny. Okay. So the main thing is the electrodes measure the voltage differences. So what do I mean when I say electrode measure the voltage differences? So we use bipolar system here. If when we have reference electrode, okay, when we have reference electrode, then when we have ground electrode, and suppose here I have one electrode. Okay, say this is, let's say this, like, uh sorry, let's say I say this channel FP2. Don't focus on this right now. We will know what is FP2 here. So generally what we do, if I want to measure the electricity from this region, suppose FP2 channel, okay. So we measure the this electrical activity is with respect to some reference channel. So generally we take ear lobes or the central portion of the lobe, and then, so basically let me explain how does it work. Like, okay, this is the our channel from where we want to measure the electricity. Okay, another is the reference and another is the ground. So how do we set up all these things? So we measure this thing. Suppose it's voltage. Let's say voltage at ground, we measure it's V0. At this channel, let's say we call it voltage, but voltage at channel C channel. Okay, and let's say this say V reference. So we have this measurement. So let's say this is V reference with respect to ground, and then we have this measure, let's say VC with respect to ground. Then we take the difference of this. It's kind of bipolar system. So what we are measuring at FP2 channel, is the potential difference change with respect to the difference. Okay, let's not make it complicated and move on. Okay, so what is it generated? Like if we talk about a single neuron's level, you may have heard about the term action potential. Like when a single neurons fire, when a single neuron fire, we get potential generation like this. So what happened, as I told you, so if suppose we have uh, we have neurons, okay, multiple, and then cell body, then synapses, okay, simple. So we have dendrite cell, we receive neurotransmitter converted into electrical activity. Then this is the cell body, neurons of cell body of the neuron. So when it crosses the after the summation from the all the dendrites, if it crosses the threshold, so suppose in this case, in rest period, like neurons is not firing, nothing is happening if this is certain. So the voltage at that time is minus 20 millivolt. Okay, so when we receive neurotransmitter and it cross the thresholds, so it if it cross the, suppose in this case the threshold is minus 55 millivolt, so if it crosses, then neurons fire. Let's not get into what is repolarization, and what is the refractory period. Just let's only focus on this is how we get the potential at single firing. Okay. So what happen when tens of thousands of neurons start firing? So this is what we are going to study. In case of in case of EEG, we record that activity. So this is how a typically EEG signals look like. So the raw EEG signal is continuous. It is characterized by rhythmic synchronized oscillation. We also call sometimes brain waves. Okay, it is in simple terms brain waves. Okay, so you see this is suppose this is different channel. So let's say this is channel C3, this is channel C4, this is CP2, this is CPZ or something. Let's say anything. Don't focus too much on the naming of the channel. We will explain. We will see how this naming we do. How do we do this naming? Okay, and this is with the time how amplitude is varying with different. So this is the typical EEG. How does it look like? Okay. Next come, what does it mean? Like how do we get information from this EEG data? It looks messy, like it's kind of look like just some oscillations with noises and all those things. And actually, to analyze any information, we broadly, like in initial stages, we divided in some frequency bands. What do I mean by frequency band? Let me explain. Okay. So what we do, we categorize in different frequency band. So first, let's say first is delta. We call this delta. So what is the range of delta? So any signal which in the range of frequency of 0.5 hertz to 4 hertz, we call that generally delta signal. Okay. Then next come theta. So this is theta. So theta is from 4 hertz to 8 hertz. We classify that as a theta hertz. So what do I mean by theta hertz? Like in 1 second, you will see from 4 cycle to 8 cycle oscillation in each signal. Or in case of delta, it is slow rhythm. Okay. And theta is a little bit faster than delta. Then comes to alpha. What is alpha? This is alpha. Alpha is 8 to 12 hertz. So there are also other names. You may heard like mu band. This is mu and alpha same. Like when we say mu band, we are talking about 8 to 12 hertz frequency or sometimes 13 hertz frequency. And when we say alpha band frequency, we mean similar thing, 8 to 12,000 hertz. But the difference why we have two terminology, it depends on the from where we are talking about. Suppose mostly when we talk about the alpha, we generally talk about the frontal region or occipital region. But when we talk about mu band, generally we talk about the motor cortex region. So okay, don't worry about this. All those terminology we will know. Okay. Then next come the beta band. The beta band. Okay. We classify signal from 13 hertz to like 12 hertz to 30 or 13 hertz to 30 hertz signal in this range. Okay, we call this beta. And next come the gamma. Okay, so gamma generally define 30 to 100 hertz. Okay. In case of EEG, we define gamma between 30 to 30 hertz. Okay, so these change predictably, like according to the person's behavior or mental state. So this is how we analyze, like we observe the behavior of the signal in certain frequency bands, like delta band, theta band, beta band, alpha band, or gamma band. Okay. For example, suppose when I want to study about the deep sleep. Okay, deep sleep. So what do we do in case we want to know about some deep sleep? Then deep sleep is mostly dominated by the slow delta waves. Okay, so we observe the slow delta waves of uh, anyone sleeping, so we can analyze the state of the deep sleep. Whereas, like when we want to know the relaxed wakefulness state, we often see the exhibition of alpha rhythm. Okay, so means when you are in very deep sleep, you will see when we are very deep sleep, you will see delta waves like this one. So, but when you are not that much deep sleepy, like wakefulness condition, you will see theta bend. Okay, or even more wakefulness, like you will see alpha rhythms. Next. And one more example, if we can say, like, suppose we are doing some active problem solving things, like you are engaged in something, your brain is very focused on doing that thing. Then we will see faster waves, faster beta waves. We will see this kind of beta waves. Okay. So due to these ability of to monitor in real time, we use the different states of consciousness and cognitive engagement to study in EEG. EEG is an invaluable tool for researchers, like studying perception and all those things. So people generally use this kind of thing. So if you want to study about the perception of any human, how our brain perceives, we can do study based on EEG. If we want to study about the memory, how does the memory work, we can do based on EEG. We can also do analysis of language perception and language study. And EEG is also used in the diagnosis of epilepsy. It's also used in evaluation of the sleep disorder and detecting the brain damage. If some certain areas of the brain, if we see normal EEG rhythm or there is some abnormality in the EEG, we can detect based on the EEG recordings. And nowadays it's very trending, like brain computer interface. So using the EEG signal, it's possible. Like when we say about the human machine interaction non-invasively, in case the most dominantly we see brain computer interface.